Hello, this is Mr. Merrier with Noteworthy Science. Today we're going to be doing subtopic A1.2 Nucleic Acids. Under the curriculum matrix, this falls under theme A, Unity and Diversity, and level of organization of 1, Molecules. Some guiding questions to really think about as we're learning this subtopic are 1, how does the structure of nucleic acids really allow for information to be stored and heredity to even be a thing? And then secondly, how does the structure of DNA facilitate accurate replication? And while the subtopic is not going to be going into the process of DNA replication just yet, it's going to set the foundation as to how that structure will inform that ability. In standard level, there are going to be 10 learning outcomes that we're going to explore today. Let's get started. This subtopic is going to be the first of a few where we're really going into some of the foundational work of biochemistry. Specifically, we're going to be looking at biomolecules. In this one, we're going to be looking at nucleic acids. And it may surprise some students why this subtopic was not called DNA. Isn't this all about DNA? Well, DNA is certainly maybe the most famous nucleic acid, but it's not the only nucleic acid. I think RNA also gets forgotten about quite a lot, and even ATP is also a nucleic acid. So this topic is really going to be exploring the structure of nucleic acids, how they differ from each other, and how those structures allow them to do all kinds of amazing things in biology. So let's begin with DNA, maybe the most famous nucleic acid. And DNA would be considered a macromolecule. Macro comes from large, and it's a large molecule. And specifically, we can say that it is a polymer, and that prefix poly means many. And specifically, what a polymer means is that it's composed of repeating subunits or monomers. In the case of DNA, monomers are nucleotides. So in this way, we can describe DNA structure as being a polynucleotide or a polynucleic acid made up of a bunch of nucleotides all bonded together. Now the name DNA comes from the fact that it's really the name comes from its structure. Deoxyribonucleic acid. Now at the moment you might be like, what the heck does this mean? That's going to make sense in a second. So I think the first place to start is, what exactly is a nucleotide? So let's explore the actual structure of a nucleotide. So nucleotides are the building blocks of all nucleic acids, whether it's DNA or RNA. The first thing that we should go into is, what exactly are the different elements that make up nucleotides? And in the case here, it's actually 5 different: carbon, oxygen, hydrogen, nitrogen, and phosphorus. Now, nucleotides are made up of 3 major components. The first component is going to be that of a phosphate group. Now, notice that I say phosphate and not phosphorus. What exactly is the difference here? Well, phosphorus is the name of this atom right here, represented by a capital P on the periodic table, but a phosphate is when that phosphorus is bound to 4 other oxygens. So it is a polyatomic ion, and in this case, that ion is bound to a sugar. So this is why we call it a phosphate group. Phosphates are going to become a really recurring theme in biology, where they associate with all sorts of biomolecules in order to accomplish certain activities. Now, where DNA gets its name from is this component right here, and that is the sugar component. So this sugar component is what we can refer to as a pentose. Now, pent, you should know from geometry, pent as a prefix means 5. And os as a suffix usually refers to carbohydrates. So this is a carbohydrate. It is a pentose. Now, a common misconception, I think, is quite reasonable maybe to assume that pent refers to a pentagon shape, but that is in fact not true. Pent refers to how many carbons make up this. And notice that this ring is not composed entirely of carbons. We have 1, 2, 3, 4, and the 5th carbon is actually outside of this ring structure. This top one is actually an oxygen. Now, specifically, this pentose is a deoxyribose, and this is where DNA gets its name from, deoxyribonucleic acid. Now, to be honest, in terms of biochemistry, this is a really odd name, deoxyribose. What does that mean? Well, D means a removal of. So one less oxygen ribose. So I guess really to understand what a deoxyribose is, we should first talk about what a ribose is. Here is a ribose. Now you might say, this looks almost exactly the same, and that's true. They are almost identical sugars with one key exception. Notice that in a ribose, there are two hydroxyl groups coming off the second and third carbon respectively. However, in DNA, a deoxyribose has one hydroxide less. I believe the naming comes from the fact that ribose was discovered first, and then when deoxyribose was discovered, it's like, oh, I guess it's the same -1 oxygen. So that's where it gets its name from. So far we have a phosphate group, we have a deoxyribose, and what exactly is this group up here? Well, this group is what's referred to as a nitrogenous base. Nitrogenous, I mean nitrogen is in the word in its entirety, meaning it's composed of nitrogen, and a base refers to its function as a chemical. And so a chemical base refers to the fact that in solution, it typically accepts protons as opposed to acids, which are proton donors. So these groups act as chemical bases. Now, this is going to be a slight difference from phosphates and deoxyribose. In every single DNA molecule, you, me, a plant, a bacteria, every single DNA molecule, every single DNA nucleotide will be composed of the exact same phosphate and deoxyribose. So why is it that we have such variety? It all has to do with the nitrogenous bases. There's in fact 4 different options for nitrogenous bases. 4 different nitrogenous bases are as follows. First, adenine, or represented by a capital A, guanine, represented with a capital G, cytosine, represented with a capital C, and thymine, represented with a capital T. Now, if you take a quick look at these structures, you should notice a few things. First of all, adenine and guanine are actually composed of 2 fused rings, whereas cytosine and thymine are composed of a single ring. In this way, adenine and guanine are physically larger than cytosine and thymine. Now, another key important aspect of nitrogenous bases is that they are able to interact with each other, and specifically, they're able to form hydrogen bonds. If nitrogenous bases are able to form hydrogen bonds with each other, what can you deduce by their chemical nature? Molecules that can form hydrogen bonds with each other are polar. So all of these nitrogenous bases are in fact polar, and because they have slightly positive areas, slightly negative areas, and they can interact forming intermolecular hydrogen bonds. However, the way in which they form hydrogen bonds is very, very specific, and something that we're going to explore in a second called complementary base pairing. Now, before I talk about that, I think it is worth noting that it's the order or the sequence of these nitrogenous bases that results in all the variety of life on the planet. So specifically, the order of these nitrogenous bases, that is what is different between you and I and a bacteria and a plant and a mushroom, etc. We all have the same phosphate and deoxyribose, but the order of our nitrogenous bases can vary slightly. And that order is something that we call the genetic code. This is what makes individuals unique from each other. This polynucleic acid right here, you can read this or abbreviate this as TGACACTG. We would not have to say the rest here because this is implied to be the same every single time. Now, looking at a structure like this appears to be quite complex, and the learning outcome does ask you to be able to draw a nucleotide. However, you are not expected to draw a nucleotide to this level of detail. You actually only need to represent it in a much more simplistic way. So let me show you how to represent a nucleotide in a very simplistic way for an exam. So when drawing a nucleotide, we're really just going to use 3 different shapes: a circle, a pentagon, and a rectangle. So the circle represents the phosphate group. This is going to be attached to a pentagon. Like so, and that pentagon is going to be attached to to a rectangle. Now, notice that my drawing slightly differs from the drawing that I have in my notes and what the IB recommends, but this is actually my preferred way of drawing. Can you predict why I included a bend? That bend represents a carbon. In fact, here's 1, 2, 3, 4, 5. Sometimes you may even see a nucleotide represented this way, with an oxygen at the top, really to emphasize the point that this is not carbon, but an oxygen instead. Now, again, you do not need to include this bend, you do not need to include this oxygen. But let me just show you how we can label this now. I would also probably include the name of any nitrogenous base of your choice. So let's just put thymine. And this would be an adequate representation of a nucleotide. So as we mentioned before, DNA is a polynucleic acid. So how does one nucleotide bind to another nucleotide? Now, this chemical reaction and how different monomers bind to polymers and vice versa is going to be explored in detail in subtopic B1.1. However, it is worth noting in this subtopic as well, because I want us to understand how different DNA and RNA molecules can actually be built and broken down. You can effectively categorize metabolic reactions in two major groups: anabolism and catabolism. Anabolism is the building of smaller to larger. So in this sense, you would get a monomer plus a monomer gives you a dimer, whereas catabolism would be the reverse. Catabolism would be a dimer or a polymer being broken down into monomers. So how does this actually happen? Well, anabolic reactions occur through something called a condensation reaction. Now, what does this actually mean? It's kind of a misnomer because condensation as an actual word typically in science refers to a phase change, something from vapor to liquid. That is not exactly what's going on here. Condensation comes from the fact that water is going to be produced as a byproduct. So let's explore how different nucleotides can be bound together using condensation reaction. So I have one nucleotide and another nucleotide. Now, nucleotides join with each other in what's called a phosphate sugar backbone. So this means that the deoxyribose is going to bind to the phosphate group of an adjacent nucleotide. So what's going on here is a condensation reaction produces water as a byproduct. Well, where does that water come from? Well, it comes from right here. If I remove this hydroxyl group and this hydrogen, you're going to be left with a residual oxygen. This is what's binding one to the next. So let's draw the result. So here's what occurred. This deoxyribose group has been bound to this phosphate group. Now, this bond specifically is a covalent bond, and it is quite strong. It also goes, its proper name is, it's a specific kind of covalent bond, something called a phosphodiester bond. Now, if a polynucleotide were to be broken down, then effectively we can do the reverse. If we do the reverse, then it means I'm going to use water to break this bond back down. The name of this chemical reaction is something called hydrolysis. Means hydro, water, lysis, split. So it literally means I am splitting with water. So this water molecule is going to be used to break apart this phosphodiester bond, and what you're left with is two independent nucleotides. Here is what I've drawn in more detail right here. Here's the phosphate group, the deoxyribose, and a nitrogenous base. And notice that the hydroxyl group of deoxyribose is binding to the next phosphate right here, and this is what's forming a phosphodiester bond. Now the name phosphodiester bond, you don't need to necessarily know what the true details of this are in this course, but in organic chemistry you likely will study ester bonds. A phosphodiester bond means that phospho, here's my phosphorus, and I have one ester and another ester. So this is where that name comes from. So when we actually explore the structure of DNA, I mentioned earlier that the nitrogenous bases bind in a very particular way. That being that they won't just bind with anything. In this case, it's something called complementary base pairing. So adenine is only going to form hydrogen bonds with thymine. And cytosine is only going to form bonds with guanine. Specifically, they are going to form hydrogen bonds with each other. If you try to include adenine to adenine or adenine to cytosine or any other combination, what ends up happening is that the polar sections do not align properly, or they may be able to form some bonds, but they'd be so unstable that the molecule would not be able to last in its current state. So A to T and C to G is the most stable configuration. So how exactly does DNA look? Well, DNA is actually composed of two strands. So we have two strands this way. However, the DNA strands do not face in the same orientation. While they are parallel in that they are facing the same plane, they are what's called anti-parallel, meaning that they are running, so to speak, in opposite directions. So in this sense, we can describe DNA as an anti-parallel double-stranded molecule. If we take a look at a simplistic model of DNA like right here, I have my phosphate, sugar, phosphate, sugar, phosphate, sugar, and then notice that on this side it's sugar, phosphate, sugar, phosphate, sugar, phosphate. Here in the middle are my nitrogenous bases forming hydrogen bonds with each other. Now what happens with this kind of model is DNA does not remain in this structure, it actually twists, and it will twist together in this kind of orientation, something called a twisted ladder structure or a double helix. Where does the term double helix come from? Well, this would be one helix and this would be another helix. So DNA can also be described as a double helix. Do you have to be able to draw this complex structure of a DNA double helix? No, we can represent a DNA double helix in a much more simplistic structure. So let's explore how to draw a DNA molecule. To draw a DNA molecule, I would first start with one strand. DNA binds in an anti-parallel fashion. So that means that the phosphate groups are facing up on this side. They would have to face down on this side. So let's draw what the complementary strand would look like. So notice that on this strand, the deoxyriboses and phosphates are facing in this orientation, whereas on this strand, they're facing in this orientation. So they are anti-parallel. Now, if this was adenine, that means this must be thymine. And if this was cytosine, that means this must be guanine. Now, as I mentioned earlier, these are polar nitrogenous bases. Thus, they're going to form hydrogen bonds. And from the A1.1 subtopic, we should be familiar of how to represent hydrogen bonds, and that being dotted or dashed lines. Now, the IB learning outcomes do specify that students don't need to know how many hydrogen bonds form between complementary base pairs. However, I do think it is worth showing on a diagram, and adenine and thymine form 2 hydrogen bonds with each other, whereas cytosine and guanine form 3 hydrogen bonds with each other. So in this way, cytosine and guanine are actually stronger base pair attractions than adenine and thymine. Now, if this was on a test, let's show how I would label this. I will show labeling in a different color just for simplicity.
Here would be my phosphate group. Here is my deoxyribose. You can demonstrate that this bond right here, you can either label this as a covalent bond, or you can label this as a phosphodiester bond. Over here, we can label this right here as a nitrogenous base. We can demonstrate that this is actually a hydrogen bond. And if you wanted to, you can even label a single nucleotide.
Take note how many marks a question is worth to determine how many labels you're going to include. So if we were to look at the structure now in more detail, you notice how we have phosphate, sugar, phosphate, sugar, sugar, phosphate, sugar, phosphate. These parts right here are very stable because they're all composed of covalent bonds, and this is what's called the phosphate-sugar backbone. This center part here is where complementary base pairs will occur. So notice that you have adenine and thymine lining up, forming hydrogen bonds, and cytosine and guanine lining up, forming hydrogen bonds. Now, if hydrogen bonds are truly being formed, then you should be able to deduce partial charges. If I have hydrogen and oxygen here, this will be slightly positive, slightly negative. Here's my hydrogen, slightly positive, slightly negative. And then over here, hydrogen, slightly positive, oxygen, slightly negative, slightly negative, slightly positive, slightly positive, slightly negative. So you can see how there's a really nice alignment between these complementary base pairs, and that's what causes the interior of DNA to be so stable. So far we've explored the structure of DNA, probably the most famous nucleic acid, but now it's time to look into the other really important nucleic acid that often gets overlooked, and that being RNA. So just like DNA, RNA is also a polymer or a polynucleotide because it's also composed of monomers called nucleotides. And in fact, RNA shares a lot of similarities with that of DNA, but a few key differences really change a lot about its overall structure and therefore function. So similarly to DNA, its name comes from the sugar that comprises its nucleic acid. Specifically, DNA, remember, was deoxyribonucleic acid, whereas RNA is ribonucleic acid. And again, you can probably already deduce that this ribo is for ribose. So let's take a look at the structure of an RNA nucleotide, and let's keep in the back of our minds similarities and differences that we find with DNA along the way. Just like DNA, it's also comprised of carbon, oxygen, hydrogen, nitrogen, and phosphorus, and its nucleotide also comprises 3 parts. The first part, the phosphate group, is actually exactly the same as it was in DNA, so nothing different over here. Now, the first major difference is, of course, its namesake. It is made up of a ribose sugar. Now, notice both deoxyribose and ribose are pentose sugars in that they are comprised of 5 carbons. 1, 2, 3, 4, 5. But the key structural difference here is that there are 2 hydroxyl groups instead of the 1 in DNA. In DNA, the group coming off of this carbon was a hydrogen and a hydrogen, whereas in a ribonucleotide, we have a hydroxyl group. So just like DNA, RNA comprises a nitrogenous base, and there are many similarities, but a key difference. Let's explore these. So just like DNA, RNA can comprise an adenine, a guanine, a cytosine, but notice that there is no thymine here. Instead of thymine, the nitrogenous base uracil is used instead. And just like thymine complementary base pairs with adenine, uracil can also complementary base pair with adenine. Drawing an RNA nucleotide effectively in this form is exactly the same as you would draw with DNA. The only difference is that you would label this pentagon as a ribose, and you cannot label this nitrogenous base as thymine. You may use instead uracil, but you could just as well use adenine, guanine, or cytosine. Now, just as DNA comprised polynucleotides, where the nucleotides are bonded phosphate sugar, RNA is the exact same. Different RNA nucleotides will bind phosphate sugar, forming these phosphodiester bonds, which would be found here. So again, a hydroxyl group coming from the ribose and a hydrogen coming from the phosphate would be removed in a condensation reaction, and the resulting bond right here would be a phosphodiester bond. So when a phosphate sugar backbone is described, Again, this refers to phosphate, sugar, phosphate, sugar, phosphate, sugar. And all of the bonds that you see here are all covalent. Thus, the structure is fairly stable. When drawing RNA, you can again draw it very similarly to DNA. The key difference between RNA and DNA is RNA is nearly always going to be single-stranded. So there will not be a complementary strand always bound to it. It's always going to be one long strand. So if it's only one strand, recall that DNA were two of these strands each connected, called a double helix. Well, RNA would be a single helix. So you can form structures that kind of look like this, or in fact, you may actually get what are called loops. Now, this structure right here might look quite interesting because it may appear like it's a double-stranded molecule, but in fact, it's still one strand. And this is really where you can start to see complementary base pairing, cytosine to guanine, adenine to uracil. So complementary base pairing, while it won't result in a double-stranded RNA structure, it can result in quite complex three-dimensional shapes and these loops. We'll look at examples of where this occurs, and maybe a very famous RNA example, tRNA. With everything that we've discussed so far concerning DNA and RNA structures and both how they're similar and different, using a table provide at least two similarities and two differences between the structure of DNA and RNA. So I've created a comprehensive compare and contrast table here. You would certainly never need to know this many on a test. But understanding how they differ is quite important in understanding how they function as well. So let's go through these. Here I've outlined a number of similarities. Both DNA and RNA are nucleic acids composed of nucleotides. The nucleotides comprise a phosphate group, which is exactly the same. The nucleotides comprise the nitrogenous base adenine, cytosine, and guanine. The backbones, or the phosphate sugar backbone, are joined by phosphodiester bonds. And the nitrogenous bases can actually form hydrogen bonds via complementary base pairing. So quite a lot in common, actually. So where exactly do they differ? Well, firstly, DNA is going to be comprising of a double-stranded structure, whereas RNA is going to be a single-stranded structure. Its namesake, DNA, is comprised of a deoxyribose sugar, whereas the nucleotide of RNA comprises a ribose. In DNA, the nucleotide comprises the nitrogenous base thymine, represented with a capital T, whereas in RNA, there will not be any thymine. Instead, there can be uracil, represented with a capital U. DNA's three-dimensional structure will be that of a double helix, whereas RNA's is slightly more variable, where you can get a single helix or loops. And while this last one is not a super good difference, it is worth noting that DNA as a polymer is on average significantly longer, in the millions of nucleotides long, whereas RNA tends to be a lot smaller comparatively, maybe in the tens to hundreds of thousands of nucleotides long. What are just some examples of nucleic acids? A lot of people think nucleic acid DNA, and that's not wrong, but I think there's a lot of other molecules that people may not realize are nucleic acids as well. So let's start with the most famous example, and that would be DNA. And this is used, why we care so much about it, is that it's this long-term genetic storage of information. So this is how all living organisms, as we'll discuss in a second, store their genetic information within a DNA molecule. And this molecule can be used to transcribe that information into proteins. Now, for RNA, there's a lot of variety here. When we discuss protein synthesis, a lot of these RNA structures will be dived into more detail. But as a quick preview, if you will, messenger RNA, represented as mRNA, is actually carrying the message of a gene from the nucleus to the cytoplasm. And so it is a complementary strand of a particular gene. tRNA, or transfer RNA, is actually bringing the amino acids to the ribosome for protein synthesis during translation. And rRNA is probably, in my opinion, the coolest of our RNAs, and yet it gets often neglected. The reason why our RNA, where its name comes from, is actually one of the structural components of a ribosome. Now, why is it so cool? It actually acts as a catalyst. This is actually what's going to catalyze all the peptide bonds in new proteins. It's kind of amazing. And lastly, it could be argued that the next most famous molecule after DNA in biology is ATP, something that we'll discuss a lot when we look at cellular respiration. Now, why am I talking about ATP here? Well, take a look at this structure. This structure should look very similar to you. Here we have a nitrogenous base, or adenine. Here, this should look familiar to you. We have a ribose sugar. And over here, we have phosphate groups, and not just one, but three, and that's where its name comes from, adenosine triphosphate. So ATP, one of the most important molecules in biology, is also a nucleic acid. Now, what's really amazing about nucleotides is their ability to store information. And so what's kind of an amazing figure is that every single living organism, from animals, plants, fungi, single-celled eukaryotes, protists, and bacteria, all of these use DNA as their genetic material, meaning all of these organisms are storing their information as genes using the DNA molecule. Now, this figure might seem hyperbolic. What about things like viruses? Some viruses actually do store their genetic material as DNA, such as bacteriophages, which infect bacteria, but many actually use RNA instead, such as very famously SARS-CoV-2, which can cause COVID, and HIV, which can cause AIDS. And so, is this not contradictory? These two actually use RNA instead of DNA. Well, something that we'll explore a lot in this course is kind of what do we mean when we say alive? And in this case, when I say we mean as a scientific community, generally do not consider viruses to be considered alive. This is a concept that we'll explore a lot in this course, but a main reason is that they effectively are not self-sufficient. They cannot carry out metabolism, reproduction, movement, any of these very important qualities without a host's help. They cannot do these things on their own, and as far as our definition is concerned in this course, they're not considered alive. But this point is going to be mentioned a lot in this course. So the key learning outcome here is all living organisms use DNA and not RNA as their genetic material. Now, a concept that we've explored already previously is that of complementary base pairing. Now, you may be wondering, we already talked about this, why talk about this more? To be honest, I did not talk about this enough. Complementary base pairing is one of the most important and crucial aspects of DNA and RNA, and hopefully at the end of today's lesson, you'll understand why that is. So as we've mentioned, complementary base pairing is very, very specific, where adenine will only form complementary pairs with thymine, and guanine will only form complementary base pairs with cytosine. And as mentioned previously, the fact that they're making hydrogen bonds should already tell you the fact that these two nitrogenous bases are in fact polar. Now, why couldn't, for instance, adenine form bonds with guanine or cytosine or perhaps another adenine? And I think this is a concept that we often personify, I think, molecules, like adenine wants to bind with thymine, or guanine doesn't want to bind with thymine, or something like that. And understand that in the chaotic mess that is biochemistry, adenine is almost certainly coming into contact with guanine or cytosine or another adenine all the time. It's not that it's choosing not to bond with it, but it won't align properly. And here's a good example of showing why that would be. If you were to try to line up an adenine and a cytosine together, and remember they would be anti-parallel, the polar groups just don't align. These two are positively charged, these two are negatively charged, and they would actually have some repulsion forces. The same thing could be true if you tried to align a guanine with a thymine. Now, if you tried other combinations, you may be able actually to get a hydrogen bond, but you will not get anywhere as stable as an adenine-thymine and a cytosine-guanine bond, or an adenine-uracil bond. Whichever the most stable configuration will be favored in a long-term structure. This is a concept that we'll explore more in additional higher level. Now, why does this matter for the DNA molecule? Well, the fact, and again, another concept that we'll look at in additional higher level, is that you have your double rings or your purines are always going to be binding to your single ring or your pyrimidines. And because of this, it causes the diameter to be constant. If this was not true, then the structure of DNA would be somewhat chaotic. You could have 4 rings across, 2 rings across, 3 rings across, and it would make the structure of DNA significantly more unstable. And so for yet another reason, complementary base pairing provides stability to a DNA structure and an RNA structure when it forms those loops. Now, the complementary base pairing nature of DNA and RNA cannot be overstated. It has huge repercussions for so many biological processes. And one of those is this little thing. If I know, if you have information on one strand of a polynucleotide, you can actually deduce its complementary strand. So here I have a DNA strand. Deduce what the complementary strand would be. You would have adenine thymine, thymine adenine, cytosine guanine, etc. Now, a typical convention when showing this is you don't have to show the hydrogen bonds, but just remember that this is where the hydrogen bonds would actually be. So when we're doing genetics, you don't actually have to demonstrate these hydrogen bonds, but keep in mind that this is where they would be. Let's imagine I have a DNA strand here. You should also be able to deduce the complementary RNA strand. Complementary base pairing, again, is not just in DNA, it also allows for RNA to actually bind complementarily. Now, I mentioned earlier that complementary base pairing is vital for biological processes. Well, let's go through some of these. One that we'll talk about in its own subtopic, D1.1 DNA replication, is this complementary base pairing nature is the core as to how DNA actually replicates itself in the process called semi-conservative replication. So here I have my original or parent DNA molecule, and what ends up happening is this molecule will split, and each of these strands will act as a template for its complementary daughter strand. So because of complementary base pairing, this code is conserved because I really only need to know one of these strands in order to deduce the other. Without complementary base pairing, this would not be possible. Another crucial biological process that relies on complementary base pairing is transcription, or the conversion, if you will, of a DNA gene to an mRNA transcript. So again, we'll explore this concept in D1.2 protein synthesis, but DNA is going to be split apart, and then only one of these strands are going to act as a template where the gene is, and a complementary mRNA strand is going to be made, thus conserving the code of this gene. This gene now, as an mRNA transcript, is transported and brought to a ribosome. And that ribosome is going to start to read this mRNA transcript in triplet codons, or three nucleotides in a row. This aspect also relies on complementary base pairing because the amino acids are bound to tRNA molecules, and through complementary base pairing, the anticodons on a tRNA are going to bind complementarily to codons on mRNA. Now, this ensures that the correct amino acid is delivered to the correct codon, thus again conserving the code. So that code was conserved throughout a DNA gene to an mRNA transcript and now a polypeptide sequence. Complementary base pairing allowed all 3 of these processes to occur. When we mention the genetic code, what we're referring to is what does each codon or triplet code represent in terms of an amino acid. So because there are 4 base pairs, ATCG or AUCG, and because the codes are in triplets, or 3, that means there's 4 raised to the 3rd power, or 64 different codons. These codons can be expressed either as a table or in a much more colorful fashion, such as something like a codon wheel. So for instance, let's imagine I have the code ACC. What does that mean? So the first nucleotide in the codon is A, the second is C, and the third one is C. Thus, ACC will actually translate ultimately to the amino acid threonine. So when we say triplet code, this is what we mean. Now, does this just apply to humans? No. This code, believe it or not, is universal. This is used, again, by all living organisms. So it's not just all living organisms use DNA, they use DNA in the same way. This means you can take a gene from a bacteria and then get it expressed by a whale or vice versa. In fact, we already do this in transgenics, where we've taken the gene of human insulin, we've inserted that into the plasmid of a bacterium, and that bacterium is able to use the same human gene because it uses the same codons in order to make insulin. So this genetic code is in fact universal and conserved across all living organisms. Is this just a neat fact? Well, yes, it certainly is a neat fact, but something that we're also going to be discussing also in this course is the fact that this is a really strong piece of evidence to suggest that all living organisms share a universal common ancestor. Or the last universal common ancestor, LUCA, L-U-C-A, is one that all living organisms are descendants from. How did I get there? Well, we have to almost look at probabilities here, and that is, what is kind of more likely? Is it more likely that this code, this exact same 64 codon code, independently evolved multiple times throughout Earth's history? Or is it more probable that this code evolved once and then it was passed on as life diverged through evolution? So we'll be using this type of argument a lot in biology. It's something called the principle of parsimony, or kind of the simplest explanation is usually the most probable explanation. So the fact that all of us share the same code is a really strong piece of evidence to suggest that we're all related. Now, finally, the last piece that I want to discuss is why DNA? Can't RNA also be used as genetic material? Why is DNA such a good, strong candidate for a genetic material? So first of all, it may seem weird to think about, there's only a variety of 4 base pairs. Isn't that limit how diverse it can be? Before we knew DNA was a genetic material, this was a point that they brought up. Some scientists did not think DNA was genetic material because it was only made up of 4 bases. However, DNA's combination is in fact effectively limitless. How can I make that argument? If there's only 4 different bases, then depending on how many nucleotides make up a polynucleotide, there are 4 raised to the n possibilities. So even if you have a polynucleotide of just 10 base pairs long, then you would have 4 to the 10 possibilities, or over 1 million different combinations possible. Now keep in mind, a single DNA molecule can be over 1 million nucleotides long. So even though there are only 4 different nucleic acids that store genetic information, it is nearly limitless how much the information can be stored in DNA, because all of these bases can effectively be placed in any order. The other thing that allows DNA to be in a very variable genetic material is that its length is not fixed. You can have DNA molecules that are very small all the way to enormous. In fact, even if you look at just the chromosomes within humans, you find that you have a really large chromosome 1, comparatively really small Y chromosome. So look at the difference between these 2 chromosomes. They differ by a large amount of nucleic acids. Now, all of this genetic information, it's good that you can store it, but it's kind of useless if it's not compressible and if it's not portable. Well, DNA as a molecule is very, 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 very thin. In fact, its diameter is just 2 nanometers. So this means that it can be compressed in a really, really, really small space, something that we'll discuss in HL. So this stat always blows my mind, because if you took all of human DNA in a single cell and you lined up DNA as a molecule, as in you took all of your chromosomes, unwound them completely, and stacked them end to end, they would be 2 meters in length. Now, all of that information is being packed in a nucleus of a diameter of 10 micrometers. That is remarkable. That compression is crazy. That's an enormous amount of information that's stored in an insanely small space. So DNA as a genetic material is very, very useful because it's highly, highly, highly compressible. So overall, this page here is really trying to get to understand why is DNA such a good molecule that all life uses as a genetic code, and what is the really importance of complementary base pairing? It's not just a neat feature, it's crucial for so many really, really important biological processes. Let's go over the learning outcomes for standard level in subtopic A1.2 Nucleic Acids. The first learning outcome is talking all about as DNA as a genetic material in all living things. And this is really getting across the point that every living thing on Earth that we know of uses DNA as their genetic material. And then kind of the rest of this topic is really understanding how are nucleic acids structured and what is their role and how does that structure enable them to carry out that role. So the first one is looking at the structure and components of the building blocks of nucleic acids, that being nucleotides, and specifically how they can be represented in this very simple circle, pentagon, and rectangle orientation. The other one is understanding how do multiple nucleotides actually join together in something called a phosphate-sugar backbone, and how are these covalent bonds are actually formed, something that will be explored further in B1.1. This learning outcome is exploring the relevancy of the order of nucleic acids, or more specifically, the order of their nitrogenous bases. The base order is really what's called the genetic code, and that is the information that is used in order to build proteins. Then we started to talk about RNA as a polymer. This subtopic is not called DNA, it's called nucleic acids. And the other major category of nucleic acids is that of ribonucleic acid, or RNA. And just like DNA, they should also be able to draw a simple RNA molecule and understand how it's formed. With respect to DNA, they should be able to explain and draw how multiple DNA nucleotides interact with each other to form a double helix, and how this can be represented again in a very simple circle, pentagon, box formation, representing both covalent bonds and hydrogen bonds. Now that we understand the differences between DNA and RNA, this learning outcome is really getting across how are these two major nucleic acids similar, but more importantly, how are they different? We then went into the actual importance of why nitrogenous bases form complementary pairs. That is not just a neat feature, it informs so many other biological processes. We then went into what makes DNA such a good actual molecule to store information, and how it allows for tremendous diversity at also being able to be compacted in such a small space. And then finally, we discussed how the genetic code is actually conserved or universal across all living organisms, and what this really means a little bit for the origin of life and how every living thing is related to each other. If you wish to learn more, which I always encourage you do, this final page is a great place to start. Here is where I detail what textbooks were used and the relevant pages that I consulted in order to create this type of lesson. Underneath, I've linked a number of simulators and interactive, which really helps explore the history and the understanding of DNA and RNA. This one is particularly relevant if you're interested in how did we as a scientific community build upon previous work in order to get to the understanding that we have today. This one details something that's explored more in HL, or Chargraff's experiment. This one goes into more about the base pairing, and this is a really nice guide going step by step through the structure of DNA. These 3D models are honestly invaluable in helping to understand such complex shapes as DNA and the base pairing, and all of these are able to be viewed in 3D space and manipulated in order to really better understand how these atoms bond together. These 3D structures are again a little bit more relevant to HL as we go into the structure of a nucleosome, but they are either way very, very cool to look at how exactly DNA is packaged within eukaryotes. Finally, if you want to do some further reading, here are 3 articles that I found really, really interesting about falsification, about hypotheses in biochem, and about really all to do with kind of the history of our understanding of getting to grips with the structure of DNA. All of them I find quite interesting and a really recommended read. I always encourage students not just to learn for the sake of a test, but really to learn for the sake of learning for enjoyment.